Recombinant DNA technology is one of the most amazing breakthroughs of the modern biological era. Now, in this technology, there is a scheme of things, a series of things that go on in order to make a recombinant DNA technology successful. In this video, we are going to understand the complete overview of how a recombinant DNA technology takes place. So, without wasting any more time, let's get started. So what basically happens in a recombinant DNA technology? In this process, what we do is we choose a target DNA. A target DNA means a DNA of our choice, our desirable DNA, from which we can extract a certain type of protein or enzyme that we need. So the DNA that we want to choose, or a gene that we want to choose, which will be a part of a DNA, is our target DNA. This target DNA will be put inside a vector. Now, what is a vector? A vector is a vehicle which will allow this target DNA to move into a cloning cell. This can be a bacteria or an yeast. Inside that cell, that vector will multiply itself. It will replicate and it will multiply. On multiplication of those cells, we will have more number of target DNA. Starting from one target DNA, we can create thousands and millions copies of that target DNA, and as a result, we can get our desired product. So we need to clone the cell in order to have this recombinant DNA technology successful. So now let us look into the process step by step. The first thing we need is a target DNA, as mentioned before, a target DNA. This target DNA will be part of, suppose, any organism. It can be human or it can be any other organism. Suppose, for example, we need a particular DNA, suppose insulin DNA, the DNA that produces insulin enzyme. Now, in order to produce this protein, insulin, there has to be a gene inside the DNA. Suppose this is the human DNA. Inside this DNA, there has to be a sequence. This particular sequence, suppose it codes for insulin enzyme. Now, suppose I as a biotechnologist, I want to take out the insulin gene and create multiple copies of this. So how do I do it? Using cell-based cloning. Now you must remember that recombinant DNA technology can be of two types. Either it can be cell-based or it can be non-cell based. By non-cell based, we have a technology called the PCR technology, the polymerase chain reaction. Now this PCR technology is a completely different set of techniques that move into the PCR. So we'll discuss about PCR in a different video. In this video, we are going to consider only the cell-based DNA recombinant technology. So we want this DNA. So this insulin becomes our target DNA. Okay, the insulin is our target DNA. We need to take this DNA out, and what we will do, we need a vehicle. As I said, I need a vector. Now this vector, it can be any secondary gene or secondary DNA that is present inside bacteria. For example, I'll take one example of E. coli, Escherichia coli. Inside E. coli, we have something called as plasmid, which is a circular DNA. And it's a secondary DNA. It contains different types of genes and DNAs to protect the E. coli. Now, this plasmid is a circular DNA. Obviously, double-stranded circular DNA. This is our plasmid. So, what is our plasmid doing? The plasmid is acting as a vector. Okay. So, inside the vector, we'll place this target DNA. Now, obviously, you are trying to understand that if this is the whole DNA, from this we need, from here, we need to take out the insulin gene. How do we take it out? We need some sort of enzyme to cut this, right? We have a full strand. From this we need a part, so I need to cut my DNA. For cutting our DNA, we need enzymes called nucleases. In this case, when you need to cut an enzyme from the middle of the sequence, not at the end, from the middle of the sequence, we need an enzyme called the restriction endonuclease. This restriction endonuclease actually comprises of a lot of endonuclease enzymes. This is a common name given to a type of enzymes which can cleave the DNA sequence at a particular position within the DNA sequence, not at the ends. It is an endonuclease, so it will cut within. Exonuclease is cut at the end. Okay, if this is a string, suppose here Reynolds is written, not being sponsored, sorry. Here Reynolds is written. I want to cut out only Reynolds. Then I will use endonuclease. If I want to cut out this cap at the end, if I want trying to cut out this part, then I'll be using an exonuclease. Okay, so here I'm using an endonuclease to cut out the DNA. Okay, I can use a variety of endonucleases like BAM-H1, then we have EcoR1. So there are a lot of endonucleases. We will study about that in our future videos. So we need a restriction endonuclease that will cut out our insulin. So after we cut it out, what we get is our target DNA. This is our target DNA that we need. This is our insulin gene. The same restriction endonuclease, whatever restriction endonuclease we use, for example, EcoR1. If I use this restriction endonuclease in order to clip this, I need to use the same restriction endonuclease to cut out the plasmid. So that it will cut out a section from the plasmid and I will have the remaining plasmid to myself. Okay, I have the target DNA. I have cut out this part and I have taken out this part of the plasmid. Now, why do I need the same restriction endonuclease to cut both of these? Because restriction endonucleases actually have a recognition site. That means they see a particular pattern and they cut only at that pattern. For example, in EcoR1, wherever it sees, wherever it can recognize a sequence of GAATTC, it will cut wherever it sees GAATTC. Where it will cut right after the G. Now obviously a DNA is a double strand, so we will have GAATTC. So it will cut here and it will cut here. So this section will be split into two parts. Okay. Now in order for us to have this section cut out and again attaching to another section here, we need a complementary sequence. The same sequence must be found in order to have complementary bonding. As a result, we need the same enzyme. If I use a different enzyme, it will cut at a different space. At a different space, the nucleotide sequence will be different. As a result, there will be no complementary bonding and I cannot attach this target DNA into the plasmid. I hope this is clear. So now that we have our insulin gene and our plasmid, we need to combine them. So for combining them, we need another enzyme which is called the ligase enzyme. We use the enzyme ligase in order to attach these two. On attaching these two, we will get something like this. This is our plasmid and here we have attached our insulin. Yes. So now this set of DNA, which is a combination of the vector and our target DNA, this is called the recombinant DNA. So we have now formed a part of the technology, the recombinant DNA technology. We now have our recombinant DNA because it is a combination, a recombination of our target DNA and the vector. Now we need to transfer this target DNA inside a host. Host means a bacteria or an yeast, which will allow this one to replicate itself. So now let us move on to our next step where we see the future of this. The next step is the transformation process. In the transformation process, what we do is we ask or we induce the bacterial cell or the yeast cell to take up the vector. So for example, there are some processes called the using of calcium chloride in order to create a sense of urgency. It seems like it is a sense of urgency for the bacteria E. coli to take up the vectors like the plasmid cells. So what we do is we take a test tube and inside a test tube or an Eppendorf, whatever we want to choose, inside a test tube we have a culture of E. coli. Here we add our recombinant DNAs. All the recombinant DNAs that we have produced just now, we'll add them here. Along with it, we will add some chemicals. For example, if we have E. coli, Escherichia coli, for them, the process that we use is we use calcium chloride. When calcium chloride and recombinant DNA along with our E. coli is added in this mixture, what happens? The calcium chloride will actually force the E. coli, it will urge the E. coli, it will induce the E. coli to take up the recombinant DNA. Now here we have many E. colis and here we have many recombinant DNAs because obviously we are not producing a single recombinant DNA. We are producing a lot of recombinant DNAs. We are allowing them to enter the cells of E. coli. in presence of calcium chloride. This is one of the process. Please don't take it as the only process. There are many other transformation processes. I'm just taking the example of E. coli, and one of the examples of E. coli is calcium chloride. So when we are adding recombinant DNA, this whole process goes on. Some of the E. coli will take up recombinant DNA. All the plasmids will not enter all the cells. Now we need to move into our next step to check which of the cells have been taken up by which of the cells have taken up the plasmid and which cells have not taken up the plasmid, because we only want the cells which have taken up the plasmid which contain the recombinant DNA. Now if all the cells are not containing recombinant DNA, then all the cells are not for my use. So now we will move into our next step, which is called the screening process. In the screening process, what we do is we take that mixture, the E. coli, the mixture of E. coli that we had along with the vectors, the plasmids. We will take that mixture and we will take a Petri plate of agar and we will stick it here, or we can have a culture where we will use we can spread it on the plates, spreading or sticking, whatever, we will spread it on the plate, liquid media, and then we will use antibiotic resistance. See, some of the plasmids contain various antibiotic resistances, genes. Now these genes will allow some of the antibiotics to not act on them. So they will prevent the action of some antibiotic on them. So we can use antibiotic resistance as an indicator to check whether a particular E. coli cell has taken up a plasmid or not. If it has taken up a plasmid, the antibiotic will have no effect on it. If it has not taken up a plasmid, then the antibiotic will have an effect on it. By understanding this, we can actually screen out which of the colonies of bacteria will contain the particular plasmid and which of them do not contain the particular plasmid. When we get particular colonies that contain the particular plasmid that we need, that is which contains our target DNA, we can take out and create a pure culture of this in a different Petri plate. These colonies here are all containing the recombinant DNA only, because we have done our screening process by using, for example, antibiotic resistance, and by using this process we have taken out all the cells that contain the recombinant DNA. Now what you need to do is we need to take up this recombinant DNA and allow the E. coli cell to increase in number, to replicate itself, so that we have more number of recombinant DNA inside each of these E. coli cells, and we can extract our product. For example, which we took insulin here, we can extract our insulin as and when required. So this is the whole process of recombinant DNA technology, the whole overview. And if you want to know more about these techniques and what are the sub processes that are involved, make sure you subscribe to the channel, because we are going to take out different videos, a series of videos on different topics of life science. And of course, the coming videos will be based on the recombinant DNA technology. So if you want to stay tuned, make sure you subscribe. You will get notified as and when the videos are uploaded. So thank you everyone for watching, and until next time, cheers.